and keep track of that chat feed and respond to your questions throughout the presentation. Instead, we'll take a look at our agenda for today's presentation. Also, with a brief introduction on the who, what, when, where, and why of the GTM NER. Then I'll go over to Lauren Flynn, our coordinator of the GTM Research Reserve Waste Recycling and Restoration Project. A brief introduction to the National Science Collaboration and the partnership that the GTM NER has established with South Florida. And to 3 graduate students at South Florida who are conducting research on the restoration and recovery of the native Olympia waste. So, starting with who we are. Not advancing. Our name is the Talado-Matanzas National Estuarine Research Reserve, but we formed it to GTM NER or GTM Research Reserve, since that's a mouthful, and are one of 28 National Estuarine Research Reserves located throughout the country. Pretty much every state with a coastline has a National Estuarine Research Reserve, except for Louisiana, Connecticut, and Hawaii. When Research Reserve was established in 1999, we were known as Guana River State Park, and NOAA designated us as a NER in 1999. For those of us who are tuning in Oregon, we're in Northeast, but for our local listeners, we're specifically located in the span about 74,000 acres from Ponte Vedra Beach to Marine, about 17 miles south of St. Augustine. Well, our system has a three-pronged approach to the work that they do. So throughout this are research, education, and stewardship. We have our 74,000 acres of land as a living laboratory. If you can think of a research project, it can be here on site. Current research projects we're doing is the system-wide monitoring program. National monitoring program that looks at different water quality, changes in biological systems, and land use changes as a long-term project. We also have a lot of different wildlife monitoring research projects and currently long-term study on sea level rise and climate change. To think of our 74,000 acres of land as a living classroom. And we offer a variety of different educational programs, both formal and informal. The education team is really to relay those research findings to the general public. We have formal programs for children ranging from kindergarten through college. We have a lot of community outreach and public education, much like the presentation you guys are listening to right now. And last but not least, our coastal training program, which targets a very specific audience of local leaders and lawmakers so that we can think about the research that's being done in our local environment so that they can make responsible management decisions. And our third facet of work is the stewardship team. And we'll be doing everything that needs to be done to keep those 74,000 acres of land in its natural beautiful state, which some of those projects might be prescribed burns, which are done on site, all invasive plant eradication, and making that land accessible to recreators. So things like creating beach crossovers, maintaining the trail system, all that important stuff. And important question, why does the GTM Research Reserve exist? To natural biodiversity and cultural resources by using the results of research and monitoring to guide science-based stewardship and education. So, I'm going to turn the floor over to Lauren Flynn, our Water Restoration Coordinator here at GTM Research Reserve.
Thanks, Kristen. As I said, my name is Lauren Flynn. I would like you to please excuse my appearance today. I actually just got in from the reef doing some training and wanted to be here today and didn't have to change between. So excuse my muddy oyster workload. I'm the coordinator for this project here at the GTM, which was initiated upon receiving a grant, Southeast Supply Resources Partnership. It's a two-year project as it is, and we are almost to date the project still, and just having really a lot of work on and a lot of community support. So a bit about how the project came to be. We have a significant coastal region along with Walnut Peninsula, which is within their boundaries along the Intracoastal Waterway. Using the shoreline technique, such as rebuilding to mitigate that shoreline or halt the shoreline erosion. Other which is to preserve certified and numerous historical and artifacts, etc. Exist on the Guam Peninsula, spanning many years, in danger also of eroding and information being lost, as well as just trying to the local population of native eastern oyster here in St. Johns County and Northeast Florida. Having issue climbing for populations, y'all that code. Luckily, knock on wood, Northeast Florida, a stable population, the finding significantly since recommend the population that are able to do things oyster rebuilding and have oyster present in the waterway and close enough in proximity that we've had wonderful recruitment to the coastal reef and really how the oysters grow. So not restrict here, a lot of hope and a lot of success in the growth and recruitment of oysters to our reef here. For this project that spans 1,075 linear feet down of the southern portion of the Guam Peninsula. The scope on the side is kind of on a map. It's a little spitball, but when my wife and I are actually out there, we were going to get that reef built. It's daunting, but we're actually halfway there. We're doing a patch reef system, meaning each the reef is 20-foot section in space to allow wave energy to have access through versus and to avoid severe significant erosion at either end, the south end of the reef. It was built in a span system. So there's 28 sections of reef, and like I said, we built so nearly half that reef. Each section of reef contains 144 beds, built in a three-layer interlocking. And the reason that we chose this methodology, there's a lot of really great and innovative ways to build is because of our high wave energy. We get a lot of water sedimentation. So it has three tiers, and it's actually 4,000 pounds of shell in each of those sections. So I think in reality, we're looking to about 78,000 pounds of oyster shell before I think about in terms of poundage, it seems just astronomical. but it has been going so great, and only because of just amazing community volunteer support. So the shell we get is from a little restaurant, so therefore it's just wonderful. And we have 5 participating restaurants, and we have volunteers who go 3 times a week, all year long, as holidays, and definitely rain or shine, and collect the shell from the restaurant or temporary in recycle bin. And the staff does a really great job in keeping it just oyster shell and getting the non-organic that could also end up in that bin. So taking that shell and bringing it back here to the nurse, it goes to the quarantine period to make pathogens, and then we bag it and they'll freeze. And here's some great photos here that couldn't pick out. The top left one shows our quarantine area that was shell bank. It shows shell that hasn't been bagged yet, and then the bag of your shell. And some up down at the site, and the size of each of those sections of reef. Again, small, some of those, but oyster shell and a lot of labor is actually proving to be a really effective system methodology for our site here and we are seeing oysters come and recruit and grow the reef. That's a concept. So I said we have about one year new project. With the time we'll be completing the reef installation. We're going to kind of put a speed up on it and try to get the reef in before spawning season starts, which our side of the country is quick approaching in April, heightened obviously not till September, but before we know it, and I want to have the substrate there ready for those stems. So we're going to all those thousands of pounds of shell down, and then we're going to supplement and finish up the project by also making alternate flora, our dominant salt marsh native that has the reef that has been depleted pretty excellent significantly in front of it. So with the theme of stabilizing that shoreline in a shoreline technique, we will also couple the oyster restoration with the planting of marsh grasses. And our hard work is done last and a slew of volunteers of coming to work here in Northeast Florida to put in just a significant number of hours and oyster and things of that nature, and really just get the work done. It's pretty amazing project here, and we're really pleased with how it's going, and looking forward to seeing where it goes from here.
Thank you very much. It's a really interesting part of having this partnership between GTMR and South Cluner, at least for me, has been kind of seeing the differences between the two oyster reef restoration projects on even the geography of the land, and just things as big as having different sea oysters, which we'll do a little bit later. But first, just some background on how GTMR came into this partnership with South Cluner. The partnership is funded through the NERD Science Collaborative, which is a program that will bring community members into the research process so that research and science being done at the NERD can be rural community, not only to inform about the state or local environment or the things that are being done to maintain and upkeep their local environment, but also to get some feedback and questions answered from the side of the community, so that the owners and the community can work together to make well-informed, responsible management decisions. So take a look at the collaborative website. We will start at our NERS national website and click the research tab, and there you'll see the NERS Science Collaborative and the United States, which I believe we need to click on, or maybe not. There we go. You can see every mark is where there is currently a NERS Science Collaborative funded project. So in Northeast Florida, at the GTM Research Reserve, we have a Planning for Sea Level Rise on Coast project funded by NERS Science Collaborative right now. And go all the way over to Oregon, Tuesday, Oregon, and we see their project, the collaborative approach to the restoration of native Olympia oysters. And our specific grounded by NERS Science Collaborative here at GTM NER focuses on relaying information and findings from NERS Science Collaborative funded project. And we chose South Flue. We've been doing oyster reef restoration for more than 3 years. We felt like they'd be a great source of information and knowledge, so we could share ideas and share our experiences and difficulties and how we've addressed those difficulties. It's really interesting up until this point. And to hear from our 3 graduate students. But first of all, just show you the different species we're working with. Here in Florida, we have eastern oyster. So this is the oyster that that Lauren's been collecting, or our volunteers have been collecting to build the reef. Maybe kind of hard to see. And it's South Flue in Oregon. These are the oysters, but the specific oyster, a very large oyster. I think it's one that South Flue is monitoring, just to make sure it doesn't take a common basis. It is not native. But the oyster that is being used for being replenished and restored in South Flue is the oyster. Well, I'll flip the thing. It's a lot of different oysters. So, without further ado, we will pass the floor over to our 3 graduate students at South Flue.
Great. Before we jump in on that, I wonder, is there opportunity for our audience to ask questions of your presenter?
Absolutely.
Okay, great. And Deborah is in our auditorium, and Deb, you want to go ahead and ask if there are any questions for the folks who come there and what we're doing there. All right. So I'm seeing in the chat box right now we may not have questions. What I'm going to do is introduce our presenter, and we'll take our questions. Okay. I recognize some of you folks from last time, and I know we had Steve Rumrill, our former research coordinator and the principal investigator on this NERS Science Collaborative grant, was our presenter. Today, we're very lucky to have 3 graduate students that are working under the auspices of that grant. And the agenda, we have them listed a little differently in order. So the first presenter here will be Mark Oates, and then he will be followed by Kate. And Kate, I'm sorry, I've forgotten your last name. Of course, Kate Pritchard, and then finally Rose Rimler. So we'll change that. But I'm going to turn over the microphone here. We're a little cup and string on this end, so Mark is going to take over my headset here, and then we'll get him into presentation mode here.
I'm going to give you a brief overview of my research on Florida or Olympia oyster reproduction here in Coos Bay. So to start, I'm going to give you a general introduction to reproduction, my research question, and then my current efforts, and then some preliminary results for you guys. Oyster reproduction is fairly straightforward, in that you have oysters that function as males that release sperm into the water, and then you have oysters that function as females that take in that sperm from the water and fertilize their eggs within their shell cavity. Olympia oysters are a little different from some oysters in that they fertilize their eggs within the shell cavity, and they actually brood the larvae for a period of 7 to 14 days, and then release the swimming larvae after that. And so that's what these larvae would look like within the shell cavity if you opened up a brooding female. So the thing that's not straightforward about oysters is that there are no true females and no true males. In fact, oysters exhibit what's referred to as sequential hermaphroditism. And what that is that they alternate what sex they are through life. And so I'll give you a brief visual of this. So all juvenile oysters mature into males as they develop, and then these males mature. They release sperm into the water, and then they immediately switch gears and become females. And these develop eggs, and these eggs are then fertilized. And once that fertilization takes place and that egg release happens, and they get ready to release larvae, they switch gears again, and they become male oysters again. And this cycle continues throughout their life for as long as the oyster continues to reproduce. So reproduction is also seasonal. It's been found to be closely tied to water temperature. So right at a temperature of 15 degrees Celsius, and above 15 degrees Celsius, this alternation of sexes happens and reproduction happens. At this temperature, both alternation of sexes and reproduction ceases. So that brings my research question. So reproduction is actually pretty well understood for the species in other places or along the west coast, but reproductive knowledge regionally for the Coos Bay estuary is pretty absent. So in order to create and craft a management strategy for self-sustaining populations for a native oyster that's on the rebound here in the bay, we need to have reproductive information available to us to inform those management strategies. Here are my research questions. I'm only going to get to 2 of them today, but the big one here is to characterize the reproductive cycle of Australurida, which is when does the reproductive season begin, when does it peak, and does it end in the estuary? How many major spawning events are there? And are these events tied to temperature, as has been found in other places? And also, is there variation within the bay? Are all the oysters following the same cycle if they're located in different geographic areas of the bay? So to answer these questions, so a study was conducted in 2012, and I went to 2 sites. This northmost star is Haynes Inlet. It's a rocky riprap in tidal environment, and the oysters are mainly settled on the rocks and other hard substrate there. This second southernmost star is Coalbank Slough, mudflat habitat. The oysters are mainly settled on pieces of wood debris that have washed up or on each other in aggregations. My protocol for 12 months, starting in January of 2012, is to collect 40 oysters per site. And so 3 of those went for a histological analysis, and 10 of those went for a condition index, and I'll get to that later. But these are all intertidal oysters. These are all about 1 foot above the mean low tide line, and they're all sampled from about the same height. So once I collected about 60 oysters per month then. Back, I measured them for length and width, and struck to them. And what I wanted to do is I wanted to remove this digestive gland area of the oyster. And the reason I did that is because this digestive gland material, and there's some gill material along the outside there, oysters have distinct gonads. They actually have a diffuse gonad that basically encircles the entire gut. So this dark material in here, that's essentially its stomach, and this white ring of tissue here contains gametes. So that's essentially oysters are eating and reproducing machines. That's basically what their body is composed of. Add this material, this nice cross section here. I put them into a wax block and hook them up to, you could call this sort of like a glorified bologna slicer. And I took a really thin, like 7-micron section of tissue, and you could put these little sections on microscope slides. You see them with a nice, pretty color, and then you can put them under a microscope so you can visualize their structures. And so here's what I did. I looked for a sex category. I'll get into that. A maturity stage to see what stage of development they were at, and egg diameters under the microscope. So first one, second oyster material is pretty easy with the stains I used because these bundles, male gametes, show up in blue, and the eggs show up in pink. It's kind of, see, these images are really yellow, but you get the idea. It's pretty intuitive. But what's not so intuitive is the fact that I mentioned before, these guys are hermaphrodites. So what I end up having is a really messy looking gonad space because female and male gametes occupy the same gonad area. So what I had to do is I had to basically classify hermaphrodites based on how much male to female gonad tissue there was. So there are varying degrees of hermaphroditism in all of my samples. So stage was, this is getting more to the point of the questions I'm trying to answer, is the maturity stage. What stage of development are their gonads in? And so I 1 through 5 scale. So 1 being they just started producing gametes. 2 means they're developing. 3, which is these images here, are just chock full of mature gametes and they're ready to spawn. And then 4 and 5 are various stages of spawning, or they're resorting their gonads basically and get rid of excess gametes. One of the preliminary results I got from looking at these stages over a 12-month period, and what you want at first is this red line on both of these graphs. This spike, red line, is telling you when gametogenesis, when they're starting to produce gametes, is happening. And for Haines Inlet, this is the major spike is detected in May, and for Coalbank Slough, the southern site, this spike happens in June. And then by that, this purple line here are stage 3 individuals which have ripe gonads. So shortly following the spike in early gametogenesis, lots of ripe individuals found throughout the sample. And these are all just male maturity stage. I have the female data yet, but it should line pretty well with this. So reproduction is kicking off in May and it's continuing through August in Haines Inlet. And for Slough, it's kicking off in June and still continuing through August. I have the September through December samples are still being processed, so this is early data right now. Before was egg diameter, and so one of these females are ready to brood. And so when you're looking at these gonad sections, you can see sizes of these egg sections, and what you can get from is at about a size of 90 to 110 microns in diameter, those are ripe eggs. And so when you start finding those, you can, these are ripe females. So what I did is I took month averages of female egg size sample, and these will only go up to June so far, but you can see this red line here is Coalbank Slough, not doing a whole lot up until up to June, but this major spike in egg diameter occurring in June for Haines Inlet, and so this is indicative that the reproductive season is beginning, and and the female eggs are starting to develop in June of 2012. And the next thing I did is I took a condition index, which is essentially telling you how much tissue there is to how much internal volume the oyster shell is. And it's telling how much mass has the oyster packed on during the year, how much meat does it have. You can kind of, because the gonad swells up so much and you have so much gamete production, you would expect it to influence the weight of the oyster during the year. So you can do sort of an indirect assessment of how the gonads are developing in these populations. And so a lot of information to take in here. Just focus your attention on these green arrows. This line is Haines Inlet, this red line is Coalbank Slough, and this green arrow shows you where mutagenesis and gonad production was first detected for both of these sites. And as you can see, after this initial detection of gamete production, there's a steady rise in body mass in the months following. And in fact, right on this peak in body mass, you start to see the red bees, which indicate that I found brooding individuals. So I opened up an oyster and larvae spilled out everywhere, which meant that they successfully reproduced and they were ready to put larvae back into the water. I don't know what some of these data are, but this initial detection, this rise in body mass would indicate that they're producing quite a lot of gonad material. Also, from that previous graph, I'm going to skip some of this stuff here. The next question is this the temperature? The answer is yes. Right around 15 degrees. This is my May collection site. 15 degrees is right around the time that they started producing gonad material, so that fits well with what's been previously. So, conclusion, when does the reproductive season begin and end? Right around May and June. Peak, August, maybe September. I still need the data in for that. Is it tied to temperature? Yes. And are oysters following the same cycle throughout the bay? They suggest there's a 1-month day at Coalbank Slough, and also they appear less healthy at Coalbank Slough than at Haines Inlet. If you notice, the red bar on that condition graph was lower than the blue bar. What does this mean? Is that Coos Bay oysters are exhibiting a cyclical summer spawn behavior, critical temperature of spawning consistent with the 15-degree literature value, and tidal populations appear to be contributing to larval supply. So thank you for your time. Are there any questions?
Well, Sir Mark. So, all right. So Mark and I have to share the microphone here a little bit. Can you hear me okay, Kristen? All right. I mean to speed you up. That's okay. You can leave that on so that you can hear the questions. Detailed physically raise your hand, we can see that you're here.
I think we're going to type it so that we can use the mute function so that we don't have the reverberation, okay?
Okay. So, Joe asked if you would mute your microphone. Great. All right. So, if we have any questions for Mark, we can move on to our next presenter and then come back at the end. Oh, we do have someone with a question. So,
I'm going to read it to you, Tom.
Oh, great.
Condition index. So, in December to March and April, prior to the dip when they start producing gametes, but they still have unexpected gametes from last season.
That's one possibility. There's a lot left over in the winter months, and it doesn't look like they've resorbed any of it up until they start spawning again. So I don't know why that dip happens. I do know, though, that they do have lots and lots of leftover gametes in winter months, and they don't seem to get rid of them. They just sort of disappear right around the time that they start producing gametes. The other question?
Between Haynes and Colmax with delay, tied to nutrient density. And
so if the delay in Coalbank Slough compared to Haynes Inlet is due to nutrients?
That's what we're asking.
I've looked at I looked at coal A concentrations in the water at both sites, and my monthly measurements have not shown that there is really a huge difference in plant food in the water for these oysters. Chlorophyll A levels are pretty consistent. In fact, they're a little higher at Coalbank Slough. I don't know why these oysters are less healthy. I'm still trying to figure that out because temperature is fairly consistent between the two sites. Salinity is lower because the Coalbank Slough site is closer to riverine inputs. But as far as chlorophyll goes, I haven't seen a huge difference, and is why there's such a difference in the health of those oysters, or 1 month delay. I'm not sure about that. All oyster species are hermaphroditic. So somebody asked if all oyster species are hermaphroditic. You have two. Yes. Yeah. In short, yes. Yes, all oyster species are hermaphroditic.
At this point.
No, they don't. But they do not fertilize themselves. Well, not we know.
Mark, I'm going to jump in here. So we're going to switch over now because we want to kind of keep on track for time. So I'm going to help Kate get set up with the next presentation, and somehow we'll have to figure a way to clap hands together for Mark so that he can hear it. But Mark, no, let's give him a round of applause, and I'll relay it to him here. Well, he's headed out to the classroom to hear it. All right, so we will now switch to our next presenter here, and that is Kate Pritchard. And so, welcome, Kate, and let me hand the microphone over to you here.
Hi, everybody. Because Mark just talked about the reproductive biology of Australurida, I'm going to be speaking about the larval supply along a marine estuarine gradient in the Coos Bay estuary. Okay, so a little bit of the biology of the species. This is, again, an estuarine species that has 3 different life stages. First is these larvae. They're planktonic, and for a few people who might not know what that means that they drift along the water, and they don't have a lot of control over their horizontal position in the water. So, for instance, they're not strong enough swimmers to swim against the current and get to a different site. Also, 2 stages of these larvae. The first is this D stage that you see in this picture that was taken by Rose, and that's characterized by a fairly ubiquitous shape. A lot of these oyster species have shapes like this. So unfortunately for all of my early stage larvae, I can't discern that this is definitely Austria-Lurida, but I counted them anyways, and I was interested in their distribution. The second stage of these larvae is an umbo stage, and that's where they develop their characteristic shape of the species. Again, these are small larvae, so they're 150 to 300 microns in diameter, and that's basically the size of a grain of sand. So they're very small. Stage of their life cycle is as a settler, and that means that they select a habitat where they want to grow and develop into these that you saw the shell of earlier. Okay, so I'm looking at larval supply throughout the Coos Bay estuary, and the Coos Bay estuary is again here along the southern coast of Oregon. And if we zoom in a little bit, that's the Coos Bay estuary. On the left here, you have the Pacific Ocean, and then all the way on the right up here is the Coos River. And so we have, I'm working with Rose, and we have 5 sites indicated by the yellow pins, and they occur along this marine estuarine gradient. And throughout the presentation, this dark blue is going to be representative of more marine sites, and the lighter blue is going to be the more riverine sites, and then there are 3 sites in between. My specific questions, I'll just be talking about 3 today. I'll mention a few that I'm going to be working on with Rose, just so you can keep those in mind. The first are larvae supplied equally throughout the estuary. And again, I like to look at early and late stage larvae, especially because I can identify the late stage larvae, and because those are the ones that are considered to be competent to settle. Second, I'm interested in if larvae are supplied to areas outside of their adult range. And in the early and late stages we'll be looking at. And lastly, I'm interested in if water quality parameters such as temperature or salinity predict larval abundance or settlement. And Rose will be talking about the settlement. But additionally, 3 other questions to keep in mind. The first is, are larvae equally likely to settle at different sites in the bay? The second is, if larvae are supplied to these areas outside their adult range, will they settle given appropriate substrate? And the last question is, does the relationship between settlement and supply vary temporally, spatially, or with temperature and salinity? Again, if you're thinking about restoration efforts, even if you put in shell bags, you want to select a site that has larval supply and successful settlement. So my methods are fairly simple. I'm using passive larval traps to look at larval supply, while Rose is using these settlement plates. And additionally, larval supply is looked at instantaneously compared to an integrated settlement time over time. So I wanted to match my time period with what the settlement plates are looking at, because otherwise you just get a 5 or 10 minute view of this larval supply base compared to a 2-week settlement plate. So these passive larval traps are basically 2-foot-high PVC tubes filled with formalin. And the way they work is that as water is carried over the top, there's an eddy that's created. Things that are alive fall into formalin, and they die and they settle out. And then we can collect them and look at them under the microscope in the lab. The first question is, are early stage larvae supplied equally throughout the bay? And I'm only going to be showing you 2 graphs over and over in this presentation. And they're going to be organized in the same way. Does that matter? On the y-axis, we have the mean abundance of either stage, and on the x-axis, we're going to be looking at dates. And these series are going to be characterized by different shapes and lines, and then along the ledger, I'm going to have the light blue to the dark blue gradient. So it's a very question, are early stage larvae supplied equally throughout the bay? And the answer is that no. Our downtown site has at least twice as many as our Hain Inlet site. Coal Bank and Catching Slough are intermediate, and there's almost no larval supply at all at Empire. So in answer to our question, that's no. Likewise, we see the same pattern of very high abundance at downtown and very low abundance in our Empire sites with intermediate sites, both at Haines Inlet, Coal Bank, and Catching Slough. It doesn't look like they're supplied, the late stage larvae are supplied equally throughout the bay. So our next question is if larvae are supplied to areas outside of their adult range and in late-stage larvae. So previous studies have looked at the populations throughout the Coos Bay estuary, and it was determined that at these 3 intermediate sites, highlighted by the orange check marks, there were adult populations close by, but at these farther sites, the Empire and the catching slough, indicated by the red X's, there weren't. So if the question is early stage larvae supplied to areas outside of their adult range, we're going to focus on the 2 sites that do have adult populations. The first is this Empire site. And we do not see hardly any larval supply. I think there's maybe 1 or 2 here. But then interestingly, this catching slough site up here has just as much larval supply as Coalbank slough. So it does appear there is larval supply to areas outside their adult range. For older stage larvae, again, there's that same pattern of low but still a little bit of supply to catching slough and really none at Empire. My next question is, do water quality parameters such as temperature or salinity predict larval abundance? And so in order to look at this, we're using these hobo conductivity loggers. So they log conductivity and temperature. We have it set every 10 minutes, and we have these at each of our sites. So they get really big data sets. Haven't really had a chance to look at them closely or really deal with them. But this is just to highlight some differences in the sites. And I'm just focused here on the first time period where there's really distinct separations between larval abundance at the sites. So this is the salinity and these box and whisker plots. And basically just what you want to note is the majority of the data are inside the boxes, 70%. And then these arrows are each 15% on either side. So it doesn't really look like the temperature and salinity predict early stage larval abundance. And the reason that I say that is because if we highlight here our Coalbank and catching slough, they have about the same number of D stage. But if you look at their salinity, there's a huge range in catching slough daily, and it can be up to 22 parts per thousand, which could be really stressful. But here there's not that much variation at Coalbank slough, and yet they still have the same number of larvae. Likewise, the down site and the Coalbank slough site have very similar salinities comparatively, but they have very different larval abundances. So it doesn't really look to me like there's a pattern there. And like for temperature, for this first time interval, they're all very similar, the temperatures across these sites. So I don't see a pattern there either. And for late-stage larval abundance, the same thing. There doesn't really appear to be any kind of pattern. Just to take a closer look, though, these sites are really very different in their temperature and salinity regimes. So, for instance, this is our catching slough. This is the most... Oh, there's a question here. Okay, I'll do that at the end. Okay, so looking at catching slough, there's this data point down here. It's our most riverine site. And our temperature and salinity loggers indicate that although temperature is very stable across this first time interval, there's, again, this huge variation in salinity, which could potentially be very stressful to oysters. And then we have our next 3 sites, Haines Inlet, Downtown, and Coalbank. And they're all very similar in temperature and salinity, and temperature and salinity seem to be very stable across time. And then likewise, the Empire sites down here where there are no larvae, they seem to be very stable with temperature and salinity again. But temperature is much lower and salinity is much higher. So it'll take some time to look at all of these data, but it could be interesting. So that's near the end. I just want to talk about the next steps a little bit. First of all, these are all preliminary data, and I'm still counting plankton samples. I need to finish doing that. Rose and I are going to be looking at this second question, that if the larvae is supplied to outside of their adult range, will they settle given appropriate substrate? And then we'll be trying to examine the relationships between settlement and supply. I think she might be talking about that a little bit. And then we really need to focus on this temperature and salinity, or what makes the sites different, larval abundance or settlement. And the question from the GTM Research Reserve. And the question is, when you refer to close by with adult oysters, what is the actual distance? I don't know. We're examining the same sites that this other paper, Steve Rumrill, looked at. There are adult populations at our sites.
I'm the question asker right here. What I'm curious about is you don't know necessarily the distance between those sites that not have adult, oysters close by. How far down the river system is.
So they sorry? Okay. They looked at just a few different sites, and they did some transects, I think, but I don't think that they encompass the whole bay. And so one of our goals out here for this summer is actually going to be to do detailed adult surveys. And that's going to be both intertidal work and subtidal, I think, using an ROV. And that could really potentially influence the number of larvae that are available to these traps. So that is something that we want to look into. And lastly, I'd like to determine the mechanisms of retention for these oyster larvae. Okay. It's interesting that at this last site down here, we didn't really we get larvae at all. And some of our preliminary data suggests that there's this point here in the middle of the bay. When the tide comes in, it doesn't have time to come past this point, and then goes back out again. So it means that all of these waters in the upper part of the bay might be retained. That's why we don't see any larvae down at Empire. It may not have to do with temperature or salinity at all. So I think that's the end. And we have one more question. How do you differentiate the effects on temperature to get production, to abundance, and temperature? Do you differentiate the effects of temperature to get to production and to... Okay, so we're supposed to be able to kind of integrate all of our results to come up with a management plan and to be able to suggest sites to restoration workers and managers. There are so many other variables, including where the adults are located and maybe some more fine-scaled chlorophyll A data or something like that, that we got. I'm not really sure that temperature necessarily is the thing to link gamete production and abundance of larvae. Does that answer your question?
My question, I guess, was because Mark talked about how the oysters won't even produce anything if it's lower than 15 degrees. So, I mean, you're not seeing a bunch of oyster larvae in the waters. I mean, how can you tell that's just not because they just didn't produce them versus effects on that?
I'm not sure. I kind of missed out on the part of your question, I think.
So my question is, I'm just wondering because talked about how oysters don't produce larvae depending on temperature. So how do you know in regards if you see the larval supplies in the water, it's just not if they didn't produce them rather than other variables, the tides or something.
Okay, so the actual question? Yeah? Okay. honestly, we don't. We have settlement data from a couple different years. No one has looked at the larval supply, but we do have the settlement data from a couple different years, and they suggest that what was kind of a trend, that they're not the lower marine sites, but also that later in the year, my later time points, in the middle of September, the settlement really starts decreasing. And that maybe because the larval supply has drained, and there's just less settlers, or there could be some kind of mortality, or so we're not sure, but it seems what we're seeing is probably a result of the larval supply, and I'm not sure exactly how that's tied to the reproductive output. Okay, we're gonna move on. Thank you.
Big round of applause there for Kate. So Kate, they're going wild. The crowd's going wild. You did a nice job. All right. So I'll introduce our third presenter and get her set up here just momentarily. This is and a little bit long here. So are you all okay with that? Or y'all okay with that in Florida? If we're a little bit long? Thumbs up. And good, and I hope our local crowd is too. Without further ado, Rose Rimler is here to talk a little bit about larval settlement. Here you go, Rose. Joy is raised a hand. Not sure what she's got, but she could maybe. Yeah, I think so. Go ahead.
A question from Joy, from the local people? You can type it to me in the chat. Okay, maybe not. Okay, I think I'm just going to go ahead. Oh, no. They're ready. Should I just X? No, doesn't matter. Okay. Okay. No. Oh, no. All right. Thanks. Okay. So my name is Rose Rimler, and like Kate and Mark, I'm a graduate student at the Oregon Institute of Marine Biology. I'm still in my second year, and so I'll be talking about my master's thesis work, which is underway. Now, I focused on larval settlement and post-settlement mortality, and how those two factors might help determine the spatial distribution of Olympia oysters in Coos Bay. So on the side of the screen, I have this sort of generalized schematic of the oyster life cycle, and I won't go into too much detail because hopefully that's already been covered, maybe exhaustively. But as Kate mentioned, the oyster larva spends a period of time swimming in the plankton before it is able to settle. At some point, it becomes competent to settle, and at that point, it has to choose a location and a substrate to settle on, and then it will metamorphose into a juvenile, and then hopefully survive and grow into an adult. On the side here, I have a map that shows the most recent, or the results of the most recent survey of adult Olympia oysters. And so my basic question is, why the oysters where they are? And I'm referring to adult oysters in that question. So the blue dots here represent oyster presence. This is the oyster presence. And that's indicate oyster absence. So those areas were surveyed and they didn't see any oysters there. And just to orient everyone again, here's the South Slough area, the entrance to the Pacific Ocean, and then this is Coos Bay, which kind of curves around this land mass. This land mass contains the cities of Coos Bay and North Bend. There's a couple of little sloughs that branch off, and here's the Coos River that enters. So as you can see, those blue dots are all congregated kind of in the upper estuarine portion, an area that we call mesohaline, which is basically mid-salty, so not super close to the river output, they're not super close to the oceanic entrance. So oysters, as you know, are a sessile animal. It means they can't move. So their settlement period is basically like their one shot at picking a space to live. but for an oyster larva to settle somewhere, it has to get there first, and that getting there, larval supply or larval dispersal. So we wanted to look at these two factors together because they're intimately related. So what we did was we chose sites throughout the bay. As I mentioned, I won't go over them again, but here they are again. So those sites were within the range of the adult oyster, and at those sites there are adult oysters present, and we're outside of the range of at least the typical range of the adult oyster. And I used unglazed ceramic tiles as settlement material. I bolted these tiles to these T-shaped holders, which are made of PVC, and I put them adjacent to Kate's traps. Obviously, we had a number of replicates at each site, and all in the low intertidal. At sites that were only accessible at the low tides every month, and even then it could be a challenge. But we wanted the traps especially to be submerged as much as possible, because the way it works is that the water goes over the mouth of the trap. Larvae that are in the water get caught trapped, so they work when they're submerged. So we chose tidal heights that were low enough that the equipment would be underwater as much as possible. And we sampled every 2 weeks. We would go out and retrieve the plates and the traps that were in the field and replace them with a second set. Take that equipment back to the lab to analyze it. And here's a tile with some oysters settled on it, but this is not representative of what I had in the field. This is actually a tile that I had in a lab setting in an aquarium with a lot of a naturally dense number of oyster larvae in the aquarium. But it's interesting that any picture I could have taken of any of our field plates, because, naturally the settlement is not nearly this dense. And a lot of these oysters are a couple of weeks old. When they first settle, like Kate said, they're about the size of a grain of sand. And these plates were 6 inches by 6 inches. And it turns out that 36 inches is a lot of area to look at if you're looking for something the size of a grain of sand. So I ended up sub-sampling each plate. And in the future, I would probably choose a smaller plate. Oh, and the house that I used had a hole drilled in the center. This one doesn't show that, but that's how they were bolted to the PVC. And results, at least in terms of settlement, on the x-axis I have the date. We put everything out starting in late July, so we began collecting it in early August, and we continue this experiment until late November. On the y-axis I have average number of settlers per 50 square centimeters. You can see we had different settlement at different sites. So we had the most settlement here at Coos Bay, downtown Coos Bay, that's circled in red. We also had a pretty high number of settlement at the Haynes Inlet site, and that's the same place that one of the two places that Mark samples, and that's in blue. It's circled in blue, and it's the blue line on the graph. We had pretty low settlement at Cold Bank Slough, although we did have some, and then we had very little at Catching Slough, which is here. Not at Empire. Oh, and again, Cold Bank Slough, where I said we had not very much settlement, that's also one of Mark's sites, just to reference everyone. It's interesting to have such low settlement at that site because when you look at Kate's data, we definitely have larvae there. So I won't go on this for too long because it's a preliminary look at what we've got so far. We haven't really been into the meat of analyzing this data, but I thought it would be interesting to show this discrepancy. The bar graphs refer to the larval supply, and that means the larvae that were found in Kate's trap. And that is reference to this left-hand y-axis. That's average number of larvae per trap. And the line graph is the same data you were just looking at on the other slide. It refers to the right-hand y-axis, average number of settlers per plate. So just at one site, this Cold Bank Slough is in green. So we did have larvae there, and so they were in Kate's traps, but we just saw very little settlement there. That's why that Mark was saying the oysters seem less healthy. So, interesting site. And it would be interesting to further characterize all these sites by further analyzing our temperature and salinity logger records and any other way we can characterize them. So, the next logical question is, once we get to a site and settle there, will they survive there and will they grow to adulthood there? So, what I'm now is I collected oysters that were naturally settled at one field site and glued them to these same ceramic tiles with an epoxy. I have 2 different size classes, both medium and small is what I'm calling them, and I measured them with calipers, their length and width, and also took their picture so that I can use ImageJ, which is a free software imaging and software, to try to calculate the surface area of the shell as they grow. And once a month, I go back to the sites and I measure them again and also take a picture of them again. This is an example of one of my plates with medium-sized oysters, and as you can see, I hope you can see it. I circled these 3 oysters, and those are 3 oysters that died in the months in between when I put them out and when I went back to check on them. So once a month, probably through the summer. I'm contemplating getting oyster from a hatchery here and doing the same thing over again, but with younger, newly settled oysters that I'll be able to getting larvae from the hatchery. So one thing I am planning to do in terms of the next experiment is to examine the possibility of gregarious settlement in a species of oyster. And gregarious settlement just refers to the phenomenon that larvae may be more likely to settle in the presence of other oysters. So for example, here's a newly settled oyster, and here are a lot of other oysters that are looking for a place to settle down. This here is the foot appendage of the oyster, and this oyster, I took a picture of it while I was on a glass slide, so it's kind of sitting around the glass slide looking for a place to settle. So the young oysters, or the oyster larvae, may be responding to the presence of other oyster spat, or they could be responding to the presence of adult oysters or shell. And that whatever cue they might be responding to could be something in the proteinaceous outer layer of the shell, or it could be something in the tissue of the oyster. So I'm planning to expose that are competent to settle in the laboratory to pieces of shell as substrate, some of which have been treated with extract from adult muscle tissue, some of which have been stripped of their protein outer layer, some of which have been seeded with other spat that have settled recently, and control where the shell hasn't been tampered with in any way. So what I hope to gain from all this? Well, actually I didn't mention why I'm planning to do the gregarious study. If oysters really do show a tendency to settle gregariously, it could just be another piece of the puzzle explaining the distribution that we see, and especially explaining the difference in oyster densities that we see across their range. And to my knowledge, no one has looked at this in the Olympia oyster, although it certainly happens with other oyster species. I believe it's shown in Chrysostrea virginica and in other invertebrates. What do I hope to gain from these studies? Well, if we discover that, and maybe we have, that the larvae are supplied to different parts of the bay differently, and if they settle with less likelihood in certain parts of the bay than in others, and then if they maybe don't thrive as well at certain parts of the bay, then we can use that information to help inform oyster restoration efforts in Coos Bay. Hopefully that information could also be applied to estuaries, and maybe our techniques could be useful for other estuaries to use. And the restoration efforts are successful, and we have a thriving oyster population in Coos Bay that could help maintain water quality and provide habitat for other, hopefully, native species. And then if the Olympia oyster is restored to a really healthy population, maybe it can be harvested again for human consumption. So maybe in Florida you guys don't know this, but the Olympia oyster, at least in the state of Oregon, is protected to the extent that it can't be harvested or eaten currently. So if that were to change, then that could potentially bring a lucrative niche fishery to Coos Bay. And I have, except for my thank yous, which are extensive because I've had help, and I'm happy to take any questions if I can figure out how to work the chat. Oh, that was easy. Okay. Have any questions? Nope. Okay.
So, give Rose a big round of applause. She can actually hear this.
Oh.
Yeah. And I thank the presenters on this end of things. We ran a bit over time there, but Kristen, thanks to your folks for hanging in over time. and to our local audience here. We're going to pull out the Wizards of Oz from behind the curtain and meet our local audience here to see if they have any questions in person, I think, here in a minute. And I also recorded today's presentation, and so I'll make that recording available to you guys as well.
Thank you very much.
Thanks.
Thank you.
Great seeing you. Look forward to seeing you next month, I hope.
Yes.
All right. Take care. Bye-bye.
Bye.
Did say, I didn't want to ask. Did yours disappear in the first place?